Hello, I'm Meredith Blackwell. I'm at the International Mycological Congress, number 11, in San Juan, Puerto Rico. And this is Anna Rosling. And she is the person we're going to interview today because she's done great things in mycology. So first, Anna, I'd like you to tell me something about your family life and how you grew up and where you went to school.
So I'm from Sweden.
Uh-huh.
And unlike most people in Uppsala, I was born in Uppsala.
Oh.
But then
You stayed there.
I stayed there. I have moved around a bit. So I lived north of Sweden, and I lived in Mozambique as a child.
Oh, wow.
Because my parents were volunteers there. Then we went north of Sweden and came back to Uppsala. So from the age of 10, I've lived in Uppsala.
Yeah, and although I knew you, it was last year, I guess, I heard on the BBC about this great man who had worked in Africa and helped people and looked at ways to improve the economy. Am I getting it right?
Yeah.
But anyway, I was so inspired by the information they gave about him, and I actually heard it on 2 different reports on the BBC, that I went to a website they had recommended. Mm. And later I find out from Hector Urbina that your father had recently died, and it was then it clicked that maybe this was your father.
That's right, yeah.
And it
so that was it, yeah.
Yeah, so when you lived in Mozambique, it was both he and your mother.
Yeah, they were both. So my dad was a doctor, a medical doctor, and my mom was a midwife at the time. And they both worked. So we lived in northern Mozambique, in Nacala, and where they had the district together with one other doctor.
Yeah.
For 2 years, 79 to 81.
Well, how old were you?
So I was 5.5 when we came there.
So quite young.
So those are my first memories, basically.
Yeah, interesting. And he continued to work in Africa with the UN, maybe.
So once we returned, he did his PhD a couple of years later, and then did lots of field work in Africa.
Okay, and so then at some point you got interested in fungi?
Yep, I was
accidentally, or
sort of accidentally. It's I was of course going to be a save the world person, like my dad, and I was trying to study economics, but it was just way too boring for me. I couldn't do it, so I switched to agronomy with a major in biotechnology, and I was going to do plant breeding and tropical crops or something.
Yeah.
But then there was this microbiology class, and Roger Finlay was giving a lecture on mycorrhiza, and I was just like, mycorrhiza.
And you hadn't known they existed?
I had not thought about them much before, but then that was it for me. So I did my master project with Torgny Unestam. He was the professor before Roger, and he was interested in weathering. So we worked on fungal interactions with minerals. And then for my PhD, I had Roger as my supervisor and worked with mycorrhizal weathering.
Okay. And then from mycorrhizae, you went to fungi that were associated with mycorrhizae, or roots anyway?
Yes, it sort of drifted away from really. The one first study we did was a field study where we looked at vertical distribution of fungal species associated with roots. And then because we wanted to do experiments, we were also isolating fungi. So this was a collaboration with Renske Landewert in Holland and with Andy Taylor. So they were morphotyping and they were setting things on culture. And I was sequencing roots and we were sequencing soil. And then many years later, I went back to look at the isolates they had generated. And that's when I found this sequence type that we also seen associated with lots of ectomycorrhizal roots. And that I thought was interesting.
Yeah.
So I started digging in that, see what it could be.
And
this was...
it had a nickname, didn't it?
It got a nickname. So first, we called it the other ascomycetes. That was basically because we knew lots of roots were associated with other, like non-basidiomycetes. And I had been, since my master's thesis, interested in these, what are the ascomycetes that associate. So I had been cloning and sequencing. This was in 2003 when I realized this was the culture we had. And then I had, I finished my PhD, I had my first child, I went for a postdoc in Berkeley. I was still interested in weathering then, and I worked with phosphorus mineral solubilization. I came back to Sweden, I had another child, and then in 2006, I think, Björn Lindahl and Katarina Idmark in our department were they had lots of sequence types that they couldn't place, so they were doing chromosomal walking, and then they realized this sequence type, it is the same as Chris Chat had described as the soil clone group 1. And that's really when we realized slowly that that's what we have in culture. So I think by 2008, so 10 years ago, that's when we were like sitting down and we're like, okay, so I have this culture, it's a new lineage, I know it occurs globally, so I should really do something about this. And then I was expecting my third child, and I was up in the north, we spent all the summers in the north, so I had a really bad internet connection, and I had to like go to the library, and like connect, because I wanted to blast sequences. And in one of the sequences matches I found Terry Porter's paper where they had described the soil clone group 1. So then I was like, okay, then I have to do something more than just that.
Yeah, she was working in Redith's lab, I guess.
Yes, she was, yeah.
Belgos.
So she and Chris Chat, they did that, characterized the soil clone group 1 and showed that this is really a group of fungi that are globally distributed.
And I think Ari Japan, did he have some too, or from the Middle West?
Yeah, so what they had was that they had done 4 different sites, I think, where they did sequencing, and then they matched it with other ITS sequences. So at that time it was maybe 10 studies. So one of them were my root tips from the north, and then one was a spore from Anne Pringle, also from Duke, I think. And there were a couple of different studies.
Okay, and nobody knew what it looked like?
No.
Okay.
And it hadn't been cultured. And there we were with the culture, and I have absolutely no background in doing like taxonomy or characterization. I was like, so what do you do? You're like, I start talking around with people and asking, and they're like, oh, you have to make it sporulate. It's like, okay, so how do you make things sporulate? And then, you give it porridge. It's like, okay, try to grow it in porridge. Like, no, you put it in UV light. Okay, no, that didn't work. So we were sort of searching around, testing different things, and we tested growing on different carbon sources, and we could see some differences. Tried to re-inoculate them on roots, and so basically like testing through to find out more about it.
Yeah. And I remember about that time I was teaching mycology class with Kathy Aim, and we were very excited because we heard that you had it. And I think you sent me some things on my phone as we were in the class, and I was able to get
because you were saying that you had these speculations. What would it look like? So I sent some photos.
And so we got them live during the class.
That's
so
cool.
It must have been very late because it was a night class. But anyway, that is really a neat story. It's one of those intriguing things when you find something that wasn't even known to exist.
No, and
never seen.
It's a fun thing too, because you have to do all these things you had no idea about how to do, and then for me it's broadened my perspectives a lot on both learning about what you need to do to name a species. Like I had no idea.
Yeah, I had a fungus like that when I was in grad school, when I first started to be a mycologist, and I had to do everything possible to try to figure out what it was. Yeah, so you had one culture that had been...
we had 6 cultures, but from the same soil batch.
Okay, and
then the ribosomal genes are identical, so we consider them as just
and they had
the
been
same.
in culture how long?
So 99, they must have been plated in 99, so it was almost 10 years when we...
Yeah, and so the other day I heard your talk, and you have a second one in culture.
Yeah, so that's actually as we were then. So I went 2010. It is to IMC in Edinburgh, because we were sequencing genes and we wanted to make a phylogeny. I'd never done that before, so I was like, okay, I just have to present whatever I have and see what people think and get help from someone who can help us. So I presented in this workshop on basal ascomycetes that Tim James organized. And then after that we started talking, and he said he would help us out with the phylogenies. So that's really together with him. The year after I moved to Indiana with my family, and then I went up to Tim and we had a 1 week where we were looking at sequences.
Yeah.
That was so much fun. The most fun you can have almost.
Yeah, it's fun to share.
And we started doing the first trees, and we saw all these sequences are from spruce roots. There's something real in this pattern, because we had all the environmental sequences that I had downloaded.
So how many do you think? A couple hundred in general?
So there's 181, I know, species hypothesis in UNITE, but then there's this recent estimate with 16,000 OTUs if you use short read archive data, so I think there's lots of them.
Okay, yeah, and the other day in the nomenclature session. Did you take part?
I was there listening, yeah.
Yeah, did you have a boat?
No, I ended up not going.
Oh.
I was going to go.
Okay.
And then I decided, no, I want to go to a field trip.
Yeah.
But no, it's a hard question, I think.
Yeah, yeah, and
I think we are going to use sequences for
Right.
sure. I just
And
think we need to think about the
that was a
formulation.
I think that was a good thing to do, so they'll, maybe they can restrict which fungi can use it.
And I think it's a very good discussion that we're having.
Yeah, yeah,
And I really liked the discussion before.
it was. And you went the other night?
Yeah.
Yeah, okay. So, you mentioned having 3 children in the
Yep.
midst of all this, and is that all?
That's all.
So you get a lot of help from the husband?
I do get a lot.
Oh,
Well,
wonderful.
we're Swedish, so I guess. We have 2.5 years between each child, and then 2.5 years later comes my science paper. So
okay.
my husband
Your
always
fourth
jokes that
baby.
it sacrificed the 4th child's life, and I show science. But
Oh, how funny. Yeah, what does he do? Does he have
he's a scientist too. He's
okay.
an agriculture historian.
Oh, good. So he knows what
He
what
realized,
you're
yeah.
doing. Yeah, that's great.
I think it's good. Yeah.
Yeah, I once spent 5 weeks at Berkeley and gave a seminar, and all of these young women wanted to have lunch with me, and I thought they wanted to talk about fungi, but it was how do you manage a private life with children and mycology? My answer was different from yours, but anyway.
Yeah, I think in
So-
Sweden it's actually pretty easy.
Yeah, there are a lot
Like
more.
life is hard with kids for anyone, but
Yeah.
at least in science you're flexible to a certain extent.
Yeah, and I always, that was my worst fear, that the person who looked after my daughter would quit or leave or
Yeah.
something would happen. So that was really a problem. So anything else that I left out that you want to mention?
Not that I think of, no.
Okay.
It was very
Well,
nice.
we're really glad to have you here in the United States,
Yeah.
although we may not always recognize it that way. And
It's
I've
been a great
enjoyed
conference.
talking.
I've really enjoyed it.
Yes.
There was not a single session I did not want to go and listen to anything.
I know.
No, so
And I've been missing sessions by sitting here, because
of course.
so. But anyway,
it's a good
this will
thing.
be fun.
Yeah.
This will be fun to have. So you'll be on YouTube in a couple days, probably. So we'll let
Sounds
you
good.
know.
Yeah.
Okay. Thank you so much.
Thank you.